Hello and Assalamualaikum. My name is Sarah N. Binti Rosli. Today, me and my team are going to present about chromatography in food industry. So as you can see here, this is my team members. First one is Cherlin Amita Anak Paros. Second one is myself, Sarah N. Binti Rosli. And third one is Nurul Fatihah Binti Elvis. So let's start with the history. In 1906, Mikhail Semenovich Servet separated plant pigments, which we know as chlorophyll, using chromatography. Because the analysis results were displayed in color along the absorbent column length, he named the new method chromatography. Graphin means to write and chroma means color.
Now, I will present about introductions of chromatography. Chromatography is separations method based on divisions or distributions of dissolved substance between mobile phase and stationary phase. First is mobile phase. It moving part and in the forms of gas or liquid that move over stationary phase. Second principle is stationary phase. Static part and solid form that is fixed in a place, column or planar. It also to absorb the component that is being separate.
Proceed to the importance of the chromatography. First one, we have quality assurance and flavor profiling. Chromatography enables ongoing food product quality monitoring, analysis of flavor and aroma, constituents, and product formulation optimization to satisfy customer preference. Second one is regulatory compliance. Chromatography offers trustworthy data that regulatory agencies throughout the world need to verify the safety and quality of food, assisting products in adhering the strength guidelines and prevent recalls or fines. For the third one, we have nutritional assessment. It facilitates precise measurement of nutrients including vitamin, proteins, and carbohydrate, guaranteeing adherence of regulation governing nutritional labeling and assisting in the creation of meals that have been fortified and enhanced. Next is the type of chromatography. We have planar chromatography and column chromatography. For the planar chromatography, when the stationary phase is held up by a flat plate or paper fibers, the mobile phase passes through it either by gravity or capillary action. So, as you can see here, this is the example of paper chromatography and thin layer chromatography, or known as TLC.
Paper chromatography is a simple method to separate substances in a mixture. First, draw a pencil line near the bottom of the paper and place a small dot of the sample on it. Next, pour some solvent into a beaker and hang the paper so the bottom just touch the solvent. Make sure the dots stay above the liquid. As the solvent rises, it carries the substances with it. Remove the paper before the solvent reaches the top and let it dry. You see spots appear, each one shows a different substances. Then, mark how far the solvent traveled. This creates a chromatography which helps us in identify substances in the sample. For thin layer chromatography, or TLC, works in similar way but use a special coated plate instead of paper and give more precious result.
Comparison between TLC and paper chromatography. Firstly, the principle behind this method is different. TLC works on the principle of absorption, whereas paper chromatography follows the principle of partition. Next, looking at the stationary phase. TLC uses a thin layer of adsorbent such as silica, gel, or alumina, which is coated into a glass or plastic plate. In contrast, paper chromatography uses cellulose filter paper that holds water as the stationary phase. For the mobile phase, both techniques typically use a solvent or a mixture of solvent to carry the sample through the stationary phase. In terms of speed, TLC is generally faster to prepare and run, while paper chromatography takes a bit more time. As we can see, this is an example for result of experiment on the separation of plant pigment using paper chromatography. The objective for this experiment to separate and identify different pigment present in plants leaf using paper chromatography. First is chlorophyll B light green and chlorophyll A blue to green, xanthophyll yellow and cateroids orange. Each pigment travel a different distance, which reflect difference in their chemical properties. For example, cateroids being less polar travel further up the paper, while chlorophyll, which are more polar, stay closer to the baseline. In conclusion, this experiment demonstrates that plant leaf contain multiple pigments, each with unique properties.
Next is the TLC plant showing the result. The origin line is marked at the bottom and the solvent front is near the top. Sample 27 is shown in green and sample 28 in blue. From the diagram, we calculate the RF value. Sample 27 had an RF of 0.82, while sample 28 had an RF of 0.79. Both values are quite high, indicating strong mobility with the solvent. This high RF value suggests that both samples are relatively non-polar. The small difference between the RF value indicated that sample 27 is slightly less polar than sample 28. This also suggests that the chemical components of both samples are quite similar. In conclusion, thin layer chromatography revealed that sample 27 and sample 28 contain compounds of similar polarity and structure. Sample 27 shows slightly higher mobility on the TLC plate, indicating it is a marginally less polar.
Now, I want to talk about principal paper chromatography. Paper chromatography, a type of liquid chromatography based on partitions or absorption. In this method, the mixture separates as it moves with a liquid mobile phase through the filter paper, and water trapped in the paper acts as the stationary phase, so the components can separate because they have different attract. So it facilitates precise measurements of nutrients, including vitamin, protein, and carbs, quaterning enhanced to regulations governing nutritional labeling and ascertaining in the rations of meal that have been enhanced. Now I want to talk about principal thin layer chromatography. Thin layer chromatography uses a plate coated with a thin layer of absorbent like silica gel. This coating is the stationary phase. Second, the sample is placed on the plate and a solvent is mobile phase was moved up the plate by capillary action. So different substances move at the different speed, causing them to separate. Second, separation depends on how strongly each component sticks to the stationary phase. So the stronger attraction means lower movement, while weaker attraction means faster movement. Lastly, after separation, the component appear as spot and can be identified using detection method. So, in conclusion, chromatography is a powerful analytical tool widely used in chemical, biological, and food science. As technology advances, modern chromatography techniques continue to evolve, providing more accurate, sensitive, and efficient results in various scientific fields.
So, as you can see here, we have a list our reference that we use for this presentation. I hope you guys can search or do your own research about the chromatography in food industry. That's all from us. Thank you.